Welcome to the NASA TOPS training in Artificial Intelligence and Machine Learning for Space Biological Sciences, where we're going to be using NASA cloud-based scientific data. We're going to start this lecture off with an introduction to Google Colab, because Google Colab is the platform we're going to be using for a lot of the coding and software and model training in this course. So if you're not familiar with Google Colab or you haven't used it before, this is a free cloud-based platform that people use for coding and for running software programs. The great thing about Google Colab is it's a collaborative environment. So users can work together, you can add other people to your notebooks, kind of like you can add people to Google Docs. Google Colab is accessible through a web browser, so nothing needs to be downloaded and no software installation has to happen on your computer. And many of the popular Python libraries come pre-installed to make getting started with coding really easy, because Python is one of the main languages that people use with Google Colab. So in this course, we're going to be using Google Colab notebooks, and we'll take a look at some examples in a second. We're going to be using them to run code for accessing data from data repositories, analyzing that data, visualizing it, looking for trends in the data, and then training our machine learning models and then evaluating their performance. And this is all going to be made really easy because we can do it through the browser without having to download any additional software. So Google Colab uses what's called the notebook format. You might be familiar with this from Jupyter Notebooks. This is a notebook is an interactive document where code, text, and visualizations can all be combined in one place. So the way that this works is notebooks consist of multiple cells. Again, we're going to take a look at this in a moment. And you can intertwine different types of cells. So for example, code cells write and execute code, whereas text cells are plain text, or they do have formatting options such as headings, bullet points, and images. So what you can do is you can write some code into a code cell, you can execute it to see the output, and then you could add a text cell underneath, kind of describing or explaining or adding notes about what you're seeing. The great thing is that the cell output is available as soon as the cell is done running. And these notebooks are self-contained and shareable. So it's almost like kind of like a digital laboratory notebook for coding. And the great thing about this is that it enables documentation and reproducibility. So you can send someone your notebook and you can say, this is the exact workflow that I used to get these results. And they can run and get show that they get the same results. So your work is reproducible. So as an example of a Google Colab notebook, this is just a very brief notebook here. You can see up at the top left, this notebook is called Welcome to Colaboratory. You can view a table of contents on the left, and this has to do with those different formatting options that you can put in the text cells. You can have headers and subheaders. So here at the very top, you can see there's an example of a text cell. So this text cell has already been edited and executed, and it looks really nice with a header and some text. And then following that text cell is a code cell where we've typed in Python, we've said print hello world, and then executed that cell by pressing the little play button here, and you get hello world printed out to the screen. And then after that code cell comes another text cell. So as you can see, these text cells here being used as explanation, as directions, and then the code cells execute, and you can see the output of that exact code right after the cell. In order to create a new code cell in a notebook, you just press this code button up here at the top. And then in order to create a new text cell, you do the same with the text button. And there are other options here that you can play around with in your free time for how to edit the different cells. So in terms of how to access Google Colab, we will be providing links to Colab notebooks at the beginning of this course that you'll run through and look at the output of the code and have some edit access. But the thing is, all users have to be signed into a Google account in order to view and execute the cells in a Colab notebook. So if you don't have a Google account, you can visit myaccount.google.com to create an account and then continue on with this course. If you're interested in doing so, new Colab notebooks, like for your independent research, can be created through Google Drive, just like a Google Docs folder. Um, so you go to your Drive, Google Drive, click on New, and then under More, you see you can create a new Google Colab notebook, and new Colab notebooks appear in a new folder in your Google Drive called Colab Notebooks. Now, what's important is in order to pull in data or other types of files to be able to view or access or analyze them inside a Colab notebook, you have to link your Google Drive to a Colab notebook. And so we will do this in the notebooks that you'll be using in this course. You run code cell with the following 2 lines. You're going to import the library drive from the Python package google.colab and then you just mount the content of your drive. So pretty simple. And then that allows you to access data found in your Google Drive. So if you were interested in analyzing some data, you could upload it to your Google Drive and then port it into a Colab notebook using this step. So with that, I'm going to jump into a short introduction to space biology. Since our topic for this course is how to use artificial intelligence and machine learning to analyze space biology data and understand the effects of spaceflight on living systems. So what is space biology? If you're new to the field, just an overview, space biology is in general the study of how living organisms respond to the hazards of spaceflight. So this could be animals, it could be humans, it could be plants, anything living that is going through space travel. And the hazards of spaceflight are many, and we kind of distill them down into the 5 that I have on the screen here. So increased levels of radiation, altered gravity, different levels depending if we're talking about the ISS, Moon, Mars, having to live in a hostile environment and a closed environment. So if you think about a capsule or a habitat or even the International Space Station, these are not environments that humans are particularly comfortable in, and so that can cause some stress. Long distance from Earth, again, depending on where you are, can be very far from Earth. Communication can be difficult, and just feeling the stress of being far away from familiar places and family and friends. And then confinement in a small space can also contribute to that stress. Each of these hazards is also linked to specific potential health consequences. So some of these you may have heard of before. So for example, radiation is linked to increased levels of cancerous cells, oxidative stress, which can also lead back to cancer, vision changes, bone loss. So for altered gravity, vision changes and bone loss as well, but then also cardiovascular immune effects, muscle atrophy, and then thinking about these hostile and closed environments, it makes you more susceptible to infection because maybe your nutrition is not that great. Mood can be altered, sleep disruption, stress. Distance from Earth obviously has to do with limited healthcare support, limited resources, limited communication. And confinement, again, this has to do with lowered fitness. Maybe you're not able to exercise as much, affects your nutrition and mood negatively, and again, sleep disruption. So each of these hazards does have to do with specific health consequences. So in order to study and try to help mitigate these health consequences and hazards, space biology field employs specific research methods. So Because sending samples into space is difficult and expensive, a lot of these studies do take place on the ground. We call these ground-based studies, where we simulate space conditions on Earth in order to investigate biological responses to individual or combined hazards. So, for example, we can use centrifuges to mimic the effects of altered gravity. We can use particle accelerators or other radiation sources to mimic radiation exposure. And sometimes these can be combined. So you can have a radiation source and a centrifuge kind of in the same experiment. There are neutral buoyancy facilities or bed rest studies to simulate weightlessness. So a lot of great and valuable data has come out of these ground-based studies. And then those can be combined also with the more rare spaceflight experiments, where investigators are able to conduct their research aboard a spacecraft, on a space station, high altitude balloons, or suborbital flights. And the key thing about space biology is in this field, we make use of model organisms. We tend not to do a lot of human research. Instead, we focus on model organisms that can tell us something about the different physiological systems that are similar to humans, that are translational back to humans, so to inform astronaut health. So this can include rodents or fruit flies, and there's a lot of plant research as well. So space biology research has been going on for decades, ever since humans were interested in exploring space. And I've just put a little timeline of the different work that was done in the different decades here. So early studies took place in the 1960s. For example, you may have heard of the first animal in space, like the dog who flew on Sputnik in the 1960s. And then there were subsequent life experiments on the smaller crude space capsules like Mercury and Vostok. And these studies were really trying to understand the effects specifically of microgravity, so altered gravity, on basic biological processes. We really didn't know what was going to happen to a human or an animal or a plant under these altered gravity conditions. So studying growth, development, reproduction, orientation, are plants even able to grow in space? So a lot of great early data came out of these decades. And then in the 1970s and 80s, we started exploring more what's happening at the cellular and the physiological level. So with the advent of the space shuttle, the space shuttle mission provided an opportunity for larger experimentation platforms. So we were able to do lots more cellular biology work, immune system, bone density assessment, and to fly more animals. And then finally, from the 1990s up to the present, as the field of biology has been advancing, so has the field of space biology in terms of the different technologies that are available. So we have key advancements in molecular biology and genomics and biotechnology. We also have international space stations such as the ISS, where a wide variety of space biology experiments take place every day using very advanced technologies. We're able to study the effects of combined microgravity and radiation, and then also looking at potential applications of biotechnology in space, 3D bioprinting and tissue engineering. And just a couple of slides here to highlight the fact that space biology is international. Just a few of the different space agencies are listed here on the slide, and the different types of research that each space agency focuses on, and the different avenues. So whether it's the ISS, whether it's high altitude balloons or satellites, each of these space agencies across the world is really contributing to our knowledge in space biology across many different fields, and contributing to that knowledge. We also focus a lot on what are the Earth applications for the research that we're doing in space. So making sure that all of the investment that we're making in space travel and in space biology is also beneficial to all of us back here on Earth. So there have been many different medical innovations, for example, that have been relevant to space biology, but have really helped the field here on Earth. So, for example, multiple therapies that have to do with, bone density, muscle, immune system, um, fundamental understanding of how our cells behave, what's happening at the cellular molecular level, what are the mechanisms of different types of disease. And one way we can think about this is that being in space tends to accelerate certain phenotypes of disease. So we can use it almost as a model of disease because the disease just happens so much faster in space. And then applied medical innovations like biomedical devices and wearables have also been innovated in space and then applied to Earth. One of the great ways that space biology research has benefited the Earth is through environmental monitoring innovations. So, for example, satellites are now used as Earth observation systems to monitor natural disasters, crop growth, agriculture, etc. And then speaking of agriculture, a lot of agricultural technology that was developed for the smaller, confined, more resource intensive areas of space travel also improve crop and water usage here on Earth. So, for example, hydroponics and aeroponics and bioregenerative life support systems for sustainable farming, urban agriculture. So that brings me to talking about where we can access some of the data that we're going to be using in this course to study these different effects of space biology. So we're going to be mainly accessing our data through the NASA Open Science Data Repository, or OSDR. And OSDR is a combination of multiple different NASA projects, which I'm showing here on the screen. So on the top left here is GeneLab, which is a data repository focused on molecular omics data, high throughput sequencing data, anything that's spaceflight or space relevant. In addition, the Ames Life Sciences Data Archive puts its data into OSDR. ALSDA is focused on higher order phenotypic data sets, physiological data, bioimaging, video, environmental telemetry data, everything that can tell us what was happening during an experiment in spaceflight. OSDR also includes the NASA Space Biology Biospecimen Sharing Program, or BSP, who is responsible for collecting these biospecimens from the different spaceflight in order to maximize scientific return. BSP also collects from associated ground investigations and includes participation from the scientific community. And then finally, OSDR includes the NASA Biological Institutional Scientific Collection, or NBISC. This is a repository of biospecimen samples that have not yet been turned into data and can be requested by investigators. So if you're interested to learn more, there's a QR code on the page, as well as URL osdr.nasa.gov/bio. So we'll be visiting OSDR to get a lot of our space biology data to train our machine learning models. And just an overview of the Open Science Data Repository and the way that you can search for data. There are multiple different search filters because there are so many different types of data in this repository. So you can search by data type, whether you're interested in looking at a study, an experiment, different biospecimen or payload, or project type. Are you interested in a spaceflight experiment, high altitude experiment, ground? There are multiple different assay types, just showing a few of these here on the screen. Amplicon sequencing, behavioral data, ChIP-seq, gel electrophoresis. So whatever you're interested in terms of data types, or you can search for multiple. Also, you can filter based on the organism that you're interested in. The majority of OSDR data is rodent, but there are human cells and some non-NASA civilian astronaut human data, plant data, etc. And then within that organism, which tissue type you're interested in studying, and then what factor you'd like to subset those data sets down to. Are you interested in a radiation exposure data set, a data set that takes place over time or has a specific treatment, altered gravity, etc. So with that, we're going to do a brief overview and tour of the NASA Open Science Data Repository. So when you find a study that you're interested in on the Open Science Data Repository, you open it up and you will come to this study page. So the study page is for OSD 48, and OSD 48 is the accession number for this study. The title here is Rodent Research 1 NASA Validation Flight, and you can tell from the title that this data set contains mouse liver, transcriptomic, proteomic, epigenomic, and histology data all in one study. So if we scroll down, we can see the description of the study gives you an overview of what this experiment was trying to find, the different factors that were studied, so spaceflight versus ground control, or the different dissection conditions. We know the organism is musculus, or mouse, and the different assays contained in this data set. In terms of the project, we can also identify information about the payload, project type, the experimental platform, so the International Space Station, sponsoring agency, NASA Center, etc., as well as the contacts, all of the investigators who contributed to this investigation. We can find information about the payload, so the experimental platform that was flown on the specific mission. And the payload identifier here for Rodent Research 1 is R1. If we click here, it will open up into an OSDR payload page for R1, Rodent Research 1. So we can find a description of the rodent research hardware system, which supported this payload, and the different experiments that have been run on this payload, different physiological systems of interest. So lots of information about this payload. This was a spaceflight payload as opposed to, for example, ground control, different hardware versions, contacts for development of this hardware and payload, and the different missions, or in this case, just one mission that this payload was flown on. We can see all the different experiments in the Open Science Data Repository associated with this payload, as well as hardware and subject and biospecimens and files. Going back to our study page here, we can also see the mission where these mice were flown. So this mission was SpaceX-4. If we click here, it would open up into a mission page on OSDR for the SpaceX-4 mission, where you can find information like dates, contact information, the different payloads that flew up into space on this mission, as well as whether there are any experiment studies or files associated with this mission. So scrolling down a little bit more in our OSD 48 study file, we can also view the different protocols for the different assay types that are included in this study. So for example, if we wanted to know how the microscopy data, the molecular cellular imaging data was collected, we could click on that protocol, and it would give us information about how the samples were labeled, how histology was performed, and then how that raw data was then processed into the user-friendly output that's available here on this page. The Open Science Data Repository provides a sample table with sample names, as well as the different information metadata variables available for each of these samples. So what was the material type here? It's liver, the different factor values for the space flight factor, so different types of controls, or was this mouse actually flown in space? And then that other factor, dissection condition, some of these samples were fresh frozen, and some of them were euthanized on the ISS. Animal source, animal sex, age at launch, how they were fed, all of these different types of variables can be found in the sample table. The Open Science Data Repository also provides an assay table where you can find information for each of the samples on each of the different assays. So the way, for example, the RNA sequencing assay was conducted on each of the samples, how quality assurance was performed, what the RNA integrity number was, how library construction and selection was performed, lots of information about each of the assays in this drop-down menu. Any associated publications with this study are listed on the page, and then the actual data is available through this browser user interface under the Files tab. So you can see all the different types of data, and these are clickable folders, so you can go in and see all the different types of raw and processed data that's available. Studies are updated from time to time, so you can view the version history here. All right, so now that we are familiar with how to identify and access space biology data, we're going to wrap up here by talking about a couple of the challenges for space biology research and how machine learning can address some of these challenges. So first of all, because flying samples into space is difficult and expensive, most of the space biology data sets will be small. So they'll have a small number of samples. We call this the small n problem. But on the other hand, these days, biological data, especially high throughput sequencing data, is often very high dimensional, thinking about, genomics or transcriptomics, which measures each of the genes that's being expressed in the cell. So we end up with data sets that are very high feature, so many different measurements, but low samples, very few samples that have been measured. So this type of data can make it more difficult to model the complexity of biological systems and their interactions in space because you only have a few samples that are replicating those interactions. The other thing is, as we just saw in this OSD48 data set, we end up with heterogeneous data types, which are very valuable. They're multimodal, and lots of different data can be collected from the same experiment, from the same samples. Um, for example, transcriptomics, bioimaging, different types of treatment variables, environmental telemetry data. And it's not always easy to combine all of that data into an interpretable picture. And then in addition, we mentioned before, research findings using these model organisms must be translatable. They have to be relevant to human astronauts. So there are multiple applications for machine learning in space biology, and we're going to start getting into these as the course goes on. One of the most key applications is biological analysis and research. So taking these data, combining them together, amplifying those biological signals and learning from those signals. This helps us with knowledge discovery, with hypothesis generation, kind of going into designing new experiments. But machine learning also has other potential applications, such as automated data management or data structure, organization, and transfer, like data is being generated in space. We transfer it to Earth in an automated fashion. Also, metadata organization and management, ensuring that all of our data is consistently organized. And then, in addition, automation of experimental payloads on flight or in situ analysis of onboard spacecraft or habitats. If data is being generated on board, how can we analyze it in an automated fashion? Now, these are just some of the applications we're going to touch on in this course. And if you're interested in some future reading as well as references for these slides, they're available here.